system with hidden parameters. Hidden means unknown. So there are known parameters, there are unknown parameters, but both are integrated together within one system, and that system is hidden Markov model. Okay. With the one that we were earlier discussing, we developed model from knowns, and then we were predicting unknowns, right? Here both are integrated into one system. So an example here is this DNA sequence CGG, AT, and so on, last is GC. What is known? The nucleotides, okay, what you observe. So sequence of nucleotides. What is unknown or hidden? Whether C is protein coding or non-coding, whether G is protein coding or non-coding. So the functional or structural characteristics of these residues or nucleotides, okay, they are not known to you. What is known is the sequence only. So both knowns and unknowns, they are integrated within one model that is called hidden Markov model, and both sequences, known sequence and hidden sequence, they are characterized by Markov chain model. Okay. So let's consider some examples to understand this. So coin toss problem. So I have a coin and I am tossing it, and there is a curtain between me and you. So I have two coins and I am picking one of the two coins and tossing, and then I am showing you the result, whether it is head or tail, and you are writing. So what you are observing is the outcome, whether head or tail. You are not able to observe which of the two coins I picked up and then tossed. Okay. So coin identity is hidden from you. What you know is the outcomes for each coin toss. Okay. Okay. So here it is. Here is how you can model this. So there are two coins, coin number 1, coin number 2. They are being tossed, and then the outcomes are being shown to you. Okay. And now I am going to ask you to model it. So there are two sequences. One is sequence of the outcomes that you know because you were recording. So first toss head, second toss tail, third toss head, fourth toss head, you are recording it. Okay. So this is a sequence of outcomes that is known to you. Okay. What is unknown is the sequence of identity of the coins that was picked and tossed, okay, each time. So maybe I picked coin number 1 first time and tossed. So I am also writing actually. So that is known to me, not to you. So I am writing 1. Second time again I picked coin number 1, so I will write 1 again. Third time I picked coin number 2, I will write 2, and so on. So this is a sequence of unknowns, because this is not shown to you. You are only shown the outcomes. So there are two sequences, sequences of knowns, sequences of unknowns. Both are characterized by hidden Markov models. Okay. So let's consider the sequence of unknowns, the hidden ones. Okay. So what are the parameters? So there are two coins. So Markov model parameters. Now you should be able to tell me. So there are two coins. So you know initial probability. Now you know transition probability. Okay. So it's very simple. Two states. State 1, which is indicating coin number 1. State 2, which is indicating coin number 2. So I am asking you to describe this using Markov chain model of order 1. So Markov chain model of order 1 means memory 1. Okay. So probability of choosing coin number 1 given that the previous choose was coin 1, or probability of choosing coin number 1 given that the previous selection was point number 2, right? So only 2 possibilities. So so you would want to now define all these probabilities. So probability of transitioning from 1 to 1, okay? So probability of 1 given 1, probability of transitioning from 1 to 2, probability of 2 given 1, given that the previous peak was 0.1, okay? Probability of transitioning from 2 to 2, probability of picking 2 given that the previous peak was also 2, okay? And probability of transitioning from 0.2 to 0.1, okay? Probability of picking 0.1 given that the previous peak was 0.2. So all these probabilities are now indicated. This characterizes the sequence of unknowns, okay? And then a sequence of knowns, which are the head and tail actually that you are noting down. So a probability of, and that is 0th order model, probability of getting head from coin number 1, okay, P1. So probability of getting tail would be 1 minus P1, okay. So they should sum to 1. From coin number 2, probability of getting head is P2. So probability of getting tail would be 1 minus P2, okay. Very good. So this is given to you, observed sequence of head and tail. This is shown to you. This is not shown to you, right? The sequence of coin identity, coin that was picked and then tossed, which coin was picked. So that sequence was not shown to you, that is unknown. Hidden Markov model will help you predict this, predict the unknown sequence given the known sequence and the model, okay? Okay, let's see how we will do this. Again, tossing 3 coins together and same thing. So when you have 3 coins that you are tossing, how many parameters will characterize this sequence using Markov chain model of order 1? How many parameters for Markov chain model of order 1? Okay, how many parameters were there for 2? For the sequence of unknowns only, how many? Only transition probability parameters. So how many? 1, 2, 3, 4. 4 parameters, transition probability parameters. So when there are 3 coins, how many parameters for 3 coins? 9. So you said 9? Right, 9 parameters. So you can see actually 9 parameters indicated here. So probability of transitioning from coin 1 to coin 1, from coin 1 to coin 2, from coin 1 to coin 3, and so on, right? So total number of transition probability parameters would be 9. And similarly, probability of, this is for the observation sequence. So probability of getting head and tail from coin 1, coin 2, coin 3, and so on. Again, same thing, you will be given the observation sequence or sequence of knowns. You are not given this, you have to predict this, okay. This is not known to you, this has to be predicted, and hidden Markov model will help you that. Okay, so here are the elements of hidden Markov model. So there is an output alphabet A. For DNA sequence, output alphabets, output. So for DNA sequence, you can say that it could be A, T, C, G, that is observed, right? A state space, a hidden state space, which is hidden from you. So let's consider simple example of protein coding and non-coding, that is hidden from you, okay? Whether a nucleotide is protein coding or is non-coding, that is not known to you. So a hidden state space could be just protein coding and non-coding. So just remove these two for simplicity, okay? You can have as many hidden states you want, but let's consider only 2 here, protein coding and non-coding, okay? A probability of transitioning between states, between hidden states. and we just learned actually probability of transitioning between coin 1 to coin 2, coin 1 to coin 3, and so on, right? So probability of transitioning between hidden states, and hidden states here are coding and non-coding. So probability of transitioning from protein coding nucleotide to non-coding nucleotide, probability of transitioning from protein coding to protein coding, and so on, right? So that is the probability of transitioning from one state to another state. And next would be output probability distribution, probability of observing an outcome from a hidden state. So probability of observing A from protein coding state. Okay. So we will try to understand further as we go to the next slides. So here it is actually probability of, let's see this one, probability of getting head from state 1, coin 1, okay, probability of getting tail from state 1, coin 1. So this is output probability distribution, okay. P1 and 1 minus P1 is output probability distribution from which state? From which state? 1, okay. Similarly, output probability distribution from state 2, okay, clear? Okay. So that's it actually, this is this. And then initial state distribution, probability of starting in a hidden state, okay. So for example, probability of starting in a protein coding state and then emitting a protein coding nucleotide. So hidden Markov model is often interpreted as emitter of symbols, okay. So there are states, hidden states, and from each hidden state symbols will be emitted. So if it is a protein coding state, it will emit these nucleotides A, T, C, or G from that state. If it is non-coding state, it will again emit these symbols, these nucleotides from the non-coding state. So this is how hidden Markov model is often interpreted. So of course, nothing is emitted here. What is important that you should understand the model. Model is being explained as emitter of symbols. So that should help you understand. So the probability of observing a sequence O1 to OK is given by this. So don't worry at the moment. I will explain in a moment. So so let's consider two sequences. The hidden sequence and observation sequence. So okay, so I will use the same notation, and I think it's not difficult to understand. So probability of hidden sequence is S, observation sequence is O. Okay. This is the joint probability of hidden sequence and observation sequence. So this is probability of, so I'm just factorizing it, probability of S multiplied by probability of O given S. Okay. So joint probability is equal to marginal probability multiplied by conditional probability. Okay. Will not go into everything, but just you can explore this further. Yeah, this is it. So joint probability can be written as a product of marginal probability and conditional probability. Okay. So let's consider PS. PS is the sequence of hidden states, and that is S1, S2, Sk, okay, because it is k symbols long, okay. And I am asking you to compute this using Markov chain model of order 1. So what should come first? So this is capital S, hidden sequence. So probability of this sequence given Markov model of order 1. So what should I write here? P is starting in S1 and then transitioning to the S2 given, and last is P S K given AK minus 1. Okay, very good. So first order model. Okay, so I wrote for this, and then probability of O given S, probability of observation sequence given the hidden sequence, and zeroth order model. Okay, so an observation sequence I would write like this: O1, O2. Okay, so DNA sequence O1, O2, and last is OK. Okay, and this is conditioned on this, right? Conditioned on the hidden state. So that's why I say that, okay, let's say that S1 is emitting O1 and then making a transition to S2, then emitting O2. Okay, and then a transition is made from Sk minus 1 to Sk after Sk minus 1 has omitted OK minus 1. Okay, and then finally Sk is emitting OK. Okay, okay. So so now how I should write this using zeroth order model? So probability of, yes, probability of
O given S when S is basically known.
no, S is unknown, S is hidden state.
Given X, what is that?
Given S is, it is conditioned on S. So S is emitting O. Okay, so this outcome is dependent on this state. Okay, that is it. So probability of zeroth order model. So zeroth order model. So probability of O1 given S1. Okay, so next would be probability of zeroth order model O2 given S2, and last would be probability of OK given Sk. Okay. Now multiply these two because you want to get the joint probability of hidden sequence and observation sequence. So multiply these two, and which is shown here. When you multiply these two and rearrange, and P is indicating the probability of transitioning between hidden states, and Q is indicating the output probabilities. Okay, both are probabilities. Okay, so if you will rearrange, you write it, and if you will rearrange, then it is something like that: probability of starting in S1, okay, and then emitting O1, which is here. Probability of emitting O1 from S1. Okay, then what happens? A transition is made to S2. So probability of transitioning, there is a mistake. Probability of transitioning to S2 from S1. So this should be S1. You can correct it. Okay. So once you are in S2, then what happens? O2 is emitted. So probability of emitting O2 from S2. So O2 given S2. Okay, and you can do like this, and when you come here, so this is basically probability of transitioning from Sk minus 1 to Sk, and then emitting OK from Sk, right? Probability of OK given Sk. Done? So you just rearrange that what I showed you, you will get this. Okay, and this is the joint probability of hidden sequence and observation sequence. Okay, and this is what is being shown here actually, only this portion shown here. So if you sum over, and actually don't worry, I will skip that. So yeah, just I just discuss this, and don't worry about this because I'm not going to come to this today. So this thing clear to everyone? Okay. So if you understand this, then you are understanding hidden Markov model. So what we want to do, we want to use this to predict a splice site in a gene from an eukaryote. Okay. So before that, actually, we would like to learn about how hidden Markov models have been used for describing prokaryotic genomes. So one of the most used hidden Markov models, the classical hidden Markov model or standard hidden Markov model, is one that emits one symbol from a hidden state. Okay. So let's consider this hidden Markov model of a prokaryotic genome, bacterial genome, where the states are shown in ovals. The observation symbols are shown in squares. Okay. So this is these three ones are coding states, and these three are the shadow states, and you can interpret this as gene on the direct strand and gene on the complementary strand. Okay. So and this is the non-coding state. So again, let's interpret this as hidden Markov model as an emitter of symbols. So if you start in a non-coding state, then a non-coding nucleotide is emitted. Okay. And then a self transition can be made to non-coding state, emitting again a non-coding nucleotide, because often non-coding regions could have multiple nucleotides, right? And then a transition could be made to gene on the direct strand or gene on the complementary strand. If a transition is made to gene states or coding states on the direct strand, then a codon will be emitted. So this is codon state 1, 2, 3. So there are 3 positions. So they are here. So it will go one by one, emitting the nucleotides of the codon. So the first codon will be emitted, and then a transition is made back to the first state of the codon, and then 1, 2, 3 again, second codon will be emitted. Once enough number of codons are emitted, then a transition will be made to the non-coding state, emitting non-coding nucleotides. Okay. And if you happen to come here, actually, so these arrows have associated probabilities. So if you happen to come here, the the codons from genes on the complementary strand will be generated. Okay. So yes. Oh, emitted. Yeah, emitted is a hidden Markov models are easy to understand when you interpret them as states that emit symbols. Okay. So the reason actually we say like this because these models are generative models. You might have heard about artificial intelligence model, generative models. So they generate sequences, which means they emit sequences, they create sequences, right? So DNA and protein sequences have been created by nature, right? But in order to understand them, we use models, and models are simplifications. Models can generate these sequences, can emit these symbols or units of the polymers. Okay, those are artificial sequences, but they can mimic natural DNA and protein sequences. Okay, so that's why when people are doing modeling, they say that we generate DNA sequences, we generate protein sequences. The model emits nucleotides, the model emits amino acids, resulting in generation of protein sequences or DNA sequences. Clear? Okay. So a standard hidden Markov model emits one symbol from a hidden state. Okay. The generalized hidden Markov model of bacterial genome can emit more than one or more symbols. Okay. For example, again, the ovals are the hidden states. So if you happen to start in non-coding state, then a non-coding sequence will be emitted. Many nucleotides of the because non-coding sequence could typically be multiple nucleotides. So non-coding nucleotides will be emitted, multiple non-coding nucleotides will be emitted according to which model of non-coding sequence? You just learned. Non-coding nucleotides will be emitted according to homogeneous or inhomogeneous Markov chain. Homogeneous, non-coding. Inhomogeneous for protein coding. Okay. So homogeneous Markov chain model of non-coding sequences, and according to the probability distribution of non-coding sequences, of probability distribution of the size of non-coding sequences. Okay. And then a transition will be made either on the gene state on the direct strand or gene state on the complementary strand. So, if a transition is made to a start state on the direct strand, then a start codon will be emitted, and then a transition will be made with probability 1 to the coding state emitting coding sequence according to which model? Inhomogeneous or homogeneous Markov model of protein coding sequences? Inhomogeneous, according to inhomogeneous and according to the length distribution of protein coding sequences in the genomes, okay, which follows a gamma distribution. So, once a gene is emitted, then a transition is made back to the non-coding state, emitting either a emitting non-coding sequence, and then a transition may be made to the gene state on the complementary strand, emitting gene on the complementary strand. Okay. So, if a transition is made here, then so this is complementary strand. So, a stop codon and then shadow of gene, okay, and then the start codon, reverse complement. Okay. Okay. So, as I said that these models can be used for generating sequences. So, hidden Markov models have been used quite extensively for generating sequences, and now artificial intelligence models are being used for generating biological sequences. You might have heard about generating models like have you heard about ChatGPT? so I think you mentioned last time. So, yeah, they have the power, actually these models have the power to generate. So, they will learn the parameters and use the parameters to generate the data. So, the hidden Markov model trained on bacterial genomes, and here actually the E. coli genome that was used for creating E. coli genome. Okay. So, model was trained on E. coli non-coding sequences and E. coli protein coding sequences. Once the model parameters were learned, then that model was used to generate artificial E. coli sequence. And this is becoming really very fascinating these days, because people are now using artificial intelligence models to generate, for example, bacteriophage sequences. Bacteriophage that can mimic natural bacteriophage, but may be more efficient in killing bacteria. For example, antibiotic resistant bacteria, and those bacteria are resistant to drugs. Can we create a bacteriophage that can kill that bacteria because the drug is not working and whosoever patient is going to die? So, you can see the power of artificial intelligence model. It can generate a bacteriophage that can be efficient in attaching to that bug that has become resistant to drug and kill it. Okay. So, people are doing research. It is a risky research. US government is quite apprehensive about this, because what happened with coronavirus, generating sequences in the lab, that could be risky also. Okay. But still, I think this is the future people are trying to do, and it is similar actually, generating sequences using models. So, artificial E. coli was generated using hidden Markov model, but one gene model was developed. So, genes were so there are over 4,000 E. coli genes. They were concatenated, and one model was trained on the concatenated sequences. Okay. So, one gene model, and there was one gene model that was trained on all non-coding sequences, and all these were integrated within the framework of hidden Markov model to generate artificial E. coli sequence. Okay. And then what was done was to look at the distribution at third codon position for GC. GC, you understand? GC content, G or C. So, that is GC3 distribution. So, GC distribution only at third codon position. Okay. So, which are shown here for all the genes. So, the distribution for all the genes is shown here, and you can see for the genuine or real E. coli, this is this one, and this one is for the artificial E. coli. So what is the difference between real E. coli and artificial E. coli? Artificial was generated by hidden Markov model. What is the difference? Look at the distribution parameters. Mu is mean, sigma is standard deviation. So can you see any difference? Yes. So what is the difference?
Percentages much higher.
Okay, so you are saying that it has more variability in the GC compared to the artificial. So which is reflected here, mean is more and less same, variability is much less for artificial E. coli compared to real E. coli, right? And this happened because one gene model was made. So, and that gene model actually captured only the major trends among all genes. Okay, so major trends were these. Okay, so most of the genes actually lie in this GC range. So they got captured by the gene model, but those which were at the outliers, so those indicated, for example, in yellow, in sky blue, and here actually purple, they got averaged out. So those signals got averaged out, they were not captured. Only those that represented the majority trend, they got captured by the one model, and this is how it is. In statistics, if you have to model using all the data given to you, then that model will capture only the majority trend in that data. The outliers may not get captured. Okay, so that is reflected here when you generated artificial E. coli. So how to address this problem? Because when you generate it, it doesn't look like a real E. coli. You want to generate a sequence that mimic real, right? Oh, sigma is standard deviation variability. So variability is less for artificial E. coli compared to the genuine E. coli. So in order to address this problem, what you would want to do? Increase the number of models. So you would want to make a model for yellow, you would want to make a model for a sky blue, for green, and so on, right? Because if you make one model, it is mostly capturing these genes that have the highest frequency in this range, in the mid range, right? So which are actually blue and green and red, and the outliers are not captured. So to address this, you would want to make distinct models for all these distinct clusters of genes within the genome, and then use those models in the hidden Markov model framework to generate E. coli again. Okay, so so here I am showing you the model using the 3 gene model. So this is hidden Markov model that used 3 gene model, but there could be as many gene models as the number of gene clusters in a genome. Okay, so and then when artificial E. coli genomes were generated again, so you see from 1 model to 3 models. So can you see it is getting better from 3 models to 9 models. So it looks like the model is able to mimic the real E. coli very well as you increase the number of models from 1 to 9, right? So which means that, okay, you have to understand the limitations of the model, and then you have to try to address using the resources that you have. So same model, but we increase the number of gene models within that framework, and then we were able to generate E. coli that could mimic genuine E. coli. Okay. So this is the power of hidden Markov generative model, including hidden Markov model, and this is the trend right now. It is happening with language models like ChatGPT, generative model, and this is also happening in the field of biology. These models are being developed for generating biological sequences. and then assessing in laboratory whether these sequences can do what they were hypothesized to do in first place. Okay, if not, then okay, feedback from wet lab, refine the model, change the model, and then check again until you get your desired sequence. Okay. Okay, so hidden Markov model can be used for generating sequences. Hidden Markov model can also be used for prediction. Okay, let's consider a simple example where you have a portion of eukaryotic gene. So eukaryotic gene has exons and okay. So is there some spray, something that can, or just water? Not sure if it will work. Oh, it is not working. He's bringing, okay. Yeah, alcohol I think should work. Oh, this may work. Oh. Oh, maybe better. Okay. I can use this, yeah. So, eukaryotic gene. So, typically, eukaryotic gene has coding sequences that are interrupted by non-coding sequences, and coding sequences are called exons. So, let's say that this is exon 1, and then this is intron, and this is exon 2. Okay. So there is a boundary between exon 1 and intron, and that boundary is called 5' splice site. Okay. Okay, this boundary is 5' splice site, and this is 3' splice site. And so let's focus only on this portion. Okay, I want to predict this. So given some sequence, so let's say the sequence is A, C, T, G, A, D, something like this. Okay, and I want to predict this boundary, and this boundary means it's a one nucleotide. Boundary here is just one nucleotide. Okay. So one nucleotide, and to the left of that boundary will be exon, to the right of that would be intron. Okay. Okay. So which is shown here. So here it is. So the model for this is shown. So only consider this portion. Okay. So the model is shown. So a start state. So these exon, intron, 5', they are hidden states. They have to be predicted. Okay, you don't know. What you know is the sequence of what is known to you, sequence of nucleotides. Okay, so you are given the sequence of nucleotides, which is this, and you are given the model. And let's see what the model is, and then you have to predict the splice site, the 5' splice site. And let's see what is the model. You are given the sequence and the model. You remember the model? Probability of transitioning between states. Okay. So probability of transitioning from E to E is 90%. Probability of transitioning from E to 5' splice site is 10%. Probability of transitioning from 5' to intron state is 100%, 1. Probability of transitioning from intron state to intron state is 90%, and then transitioning to the end state is 10%. Okay, and rest of there are 3 states. Forget about a start and end state. Other than these two, there are three states, and those states emit symbols. Start and end states do not emit symbols. Okay. So there are three states. So how many total transition probability parameters? 9. So, but those that have a value 0, they are not shown. Okay. Only those that with non 0 values are shown. Okay. So, and then, so these are the transition probability parameter values. So, output probability values. So, probability of A, C, G and T from exonic state, and these all have values 0.25. This is a toy model. Okay. So, to just help you understand, they may not be natural actually, these values. Probability of A, C, G, T from 5 prime. Okay. If you look at these values, you will tell me which nucleotide could be the boundary, could be the 5 prime splice site. G, because G has 95% probability, and A with 5%. The splice site will not have C or T. Okay, good. And the probability distribution from the I state. Okay, and the probability of starting in 5 is initial probability. The probability of starting in 5 is, sorry, the probability of starting in E is 1, right? You always have to start and then make a transition with probability 1 to E. So, you always start in E. Okay. So, this defines the parameters of the hidden Markov model. So, given this and the observation sequence, you have to predict the 5 prime splice site. Okay. So, basically you have to predict the hidden sequence. If you have predicted the hidden sequence, and hidden sequence consists of the hidden states, exonic, which is E, 5 prime splice site 5, intronic, which is I. Okay, these 3 levels only. So, basically you have to predict the hidden sequence. Okay. So, predict probability of hidden sequence given, what is given to you? Observation sequence and model, right? Okay. So, you are given these and you have to predict the hidden sequence. And let us say that this probability looks like this actually. So, probability of hidden sequence given observation, and I am going to omit M actually just for simplicity. Okay. Probability of hidden sequence given observation sequence, and here is S. Okay. And it looks like this. So, I am going to consider all hidden sequences and I will choose one that gives the highest probability of hidden sequence given observation sequence, and I will predict that sequence is the one that underlies the observation sequence. Okay. So, I have to maximize this with respect to S and identify one that has the highest value. Okay, this one. Okay. So, the problem is actually maximization of this with respect to hidden sequence. Okay. You can write as this, you can write as, tell me, this is probability of S O divided by probability of O. Okay. So, omit M. I don't want you to confuse you with so many actually. Okay. This. So, maximization with respect to S, there is no S here. So, basically you are maximizing this one, right, this one, and you just did actually, you computed the joint probability of hidden sequence and observation sequence. Okay. So, you compute the joint probability of hidden sequence, again I will show you, you did it. Let us go back and see. So, actually it is in the next, no I have not, okay. So, it is here actually, here it is. You did it, right? We did together. So this is what you have to do. You have to compute this for each of the hidden sequences. Okay? And once you have computed for all hidden sequences, then pick the hidden sequence that has the highest value of probability among all. Okay? Okay, so let's then go back and we'll compute the joint probability. Okay, here it is. So we'll compute the joint probability for each of the hidden paths, and let's assume here that we will consider only those sites as 5' splite site that has G, okay, because the probability of being a splite site is 95% for G. Okay. So, I am going to, for simplicity, I am only going to consider those sites that have G. That could be potentially a splice site. Okay. So, let us look at those. So, where is G? This one. Okay. So, this is one of the, so this one is this. So, this is a splice site. To the left of it would be exonic nucleotides. To the right of this would be intronic nucleotides. So, this one I got this one, and another G, which is this one. Okay. And then G, this one, G, this one, and then this one, and finally this one. Okay. So, how many hidden sequences? How many total? 1, 2, 3, 4, 5, 6. Okay. Very straightforward. To the left of the splice site, 5' splice site is exon, to the right is intron. So, there are total 6 hidden sequences, and out of 6 we will have to select one, and that one should have the highest probability of hidden sequence and observation sequence, right. So, did I write somewhere? I have not written, but I think I wrote here. Okay. Basically maximizing hidden sequence given observation sequence is same as maximizing the joint probability of hidden sequence and observation sequence. Okay. Okay. So, now you know how to compute given a hidden sequence. Observation sequence is given already to you. So, considering all these 6 hidden sequences, compute this and then select the one that has the highest probability. Okay. And this was done and it was found. So, this is log probability, and maximizing probability is same as maximizing log probability. Okay. So, which has the highest value among them? This one, right? This is negative. So, this one is the highest. So, this one is going to be predicted as the hidden sequence that underlies this observation sequence. And if this is the prediction, where is the splice site? This is the splice site. Okay. So, this is how hidden Markov model can help in predicting. So, the idea is to find the most likely path that underlies the observation sequence, and most likely path means the path that has the highest probability of being hidden sequence, okay, given the observation sequence and the model. Clear? So, this approach is addressed because this is computationally very demanding, using a dynamic programming algorithm called Viterbi algorithm, VITERBI. Viterbi algorithm does this. It computes the probabilities. It identifies the hidden path that has the highest likelihood among all hidden paths. Okay. The other approach is to compute for each position the probability, the posterior probability of the 3 states. So, for each position the posterior probability of being exonic, being 5' splice site, being intronic. So, let us say that at first position the probability of being exonic is 90%, probability of being 5' splice site is 0, and probability of being intronic is 10%. Then which label of these 3 will you use to label the first nucleotide? So, the labels are 5, E, I. E, because the probability of being is 90%. So, you are going to use the label that has the highest probability. Okay. So, this is how for each position you are going to label each nucleotide. Okay. So, this approach is called posterior decoding. You compute the posterior probabilities for each state, okay, and then select the label that has the highest posterior probability. This posterior decoding approach is implemented using a dynamic programming algorithm which is called forward backward algorithm. Again, I am not going to go through all these complicated algorithms, but just wanted to let you know, you should understand the concepts, but there are computational algorithms that can help in predicting the most likely hidden sequence or the states, most likely states at each of the positions. For the former, Viterbi algorithm is used, for the latter, forward backward algorithm is used. Okay, clear? Okay. So, prediction, clear? So, you would want to go through these slides again. I want you to understand how machine learning models can help in prediction. Okay. These are the basics. If you understand this, rest of the things are built on this. You should be able to understand the rest. If you don't understand this, you cannot understand the more complex models. Okay. So, go through the slides again and think yourself. I think you will be able to connect the dots. You should be. So, multiple sequence alignment. So, hidden Markov model has also been used for modeling alignment of more than 2 sequences. So, 2 sequences aligned pairwise, more than 2 multiple alignment. Okay. So, hidden Markov model can also help in describing multiple alignment. So, let us look at a very simple model of multiple alignment. So, alignment of these sequences, protein sequences, and when you look at the multiple alignment, you visualize the positions where you see conservations or variability, right. So, if you look at this, there is this position number 6 which is completely conserved, only G residue, right, only G is there. So, it is completely conserved. On the other hand, there are other positions that have variations. So, there are mutations or substitutions in other positions. Okay. So, you can build a probabilistic model for this multiple alignment, and for that you would want to consider each position. Okay. So, consider position 1 and you want to compute probability of amino acids at this position 1. So, what is the probability of E at position 1? So, how many residues are there at position 1? 1, 2, 3, 4, 5. Okay. And how many times you see E in position 1? 3. So, the probability of E would be 3 by 5. Okay. So, similarly, you will compute probabilities of all the amino acids at each position, and you can keep it in a matrix, okay, in a table. You can keep it, and then given a sequence which is S1, S2, S9, you can compute the probability of this sequence to be a member of this multiple alignment. This is representing a protein family, okay, to be a member of this family by computing the probability of this sequence given this model that you just computed. Okay. So, probability of S given M, and I would assume that this S1 is situated in position 1, it is not codon position, it is position 1 in the multiple alignment. Okay. So, S1 is situated in position 1 in multiple alignment. So, then it would be probability of zeroth order model, probability of S1 in position 1 multiplied by probability of S2 in position 2 and so on. Okay. And these values will come from the table that you just generated, right, and then you compute this. Okay. And this is here probability of S given the multiple alignment model, and you will divide this by probability of this sequence given random model. So, odds score, okay, likelihood score. So, for that you can randomly reshuffle, and then compute the probability of this sequence from the random model. Take the logarithm of this is log odds score, or you can say, yeah, just say a score or whatever. And this can help you in predicting whether the sequence is a member of this protein family represented by this multiple alignment or not. Okay, clear? Okay. So here is how hidden Markov model is used for describing multiple alignment of sequences. This is called profile hidden Markov model, and it is very popular in bioinformatics. You might have heard about PFAM models or hidden Markov models for protein families or domains. Okay. So look at this alignment, and you can see that it has blocks with no gaps, and there are regions with gaps, right? And those sites with no gaps, they are called match states. Okay. So let's consider only portions that have no gaps, and then join them, and for each position you have a match state. Okay. Match state 1 is representing the first column with no gaps. Now match state 2 is representing the second column with no gaps, and so on. Okay. Match state. Okay. So B is begin state, then match 1, and so on, and finally match N, and then end state. Okay. And now imagine that you have a sequence that has insertions. Okay. So you got a sequence and there are some insertions, which means that those residues are not represented in the multiple alignment, right? They are not represented in the multiple alignment because that is something that happened in the sequence of your interest. So you would want to model the insertion. So probability of transitioning to match state, and then from a certain match state where insertion happened, you make a transition to insertion state, and there could be insertion of multiple residues. So that's why there is a self transition. Okay, transition from insertion to insertion again, because the insertion here is emitting only one residue. So self transition to insertion, and then after enough number of nucleotides or residues are emitted from insertion state, a transition is made back to a match state. Okay. Okay. So this is how insertion is modeled. Deletion, it is possible that the sequence that you are considering has deletions, which means that there are residues which are actually present in the protein family here, but they are missing from the sequence of your interest. You would want to model it. Okay. So which is modeled here. So basically what you want to skip the match states because they are not present in the sequence of your interest because they got deleted. Okay. So suppose that these two are match states and they are absent from the sequence of your interest, they got deleted. You want to skip them. Okay. So there should be an arrow from this to this, skipping these two. Okay. So which is actually shown here by these circles, and these are called silence states. They do not emit residues, they are there to skip the residues in the multiple alignment. Okay. So if you go from this state, match state, to this circle, and then here to this state, you have skipped this match state because this match state is not present in your sequence, that got deleted. Okay. So now you integrate all these three, the match states, the insertion states, and the deletion states. This is what is the hidden Markov model for multiple alignment. Okay. And this is called profile hidden Markov model because multiple alignment of sequences is often called profile. They represent a gene family or a protein family. Okay. And they are modeled using the three states: match state, insertion state shown in diamonds, and deletion state shown in circles by the profile hidden Markov model. Okay. Okay. So here again, and I will skip same thing, but it looks more neat, profile hidden Markov model. Okay. And then you can go to PFAM, and there is a tutorial actually that either today or tomorrow you will do. PFAM is used to identify a protein family that the sequence of your interest belongs to. Okay? You have got a sequence and you want to know which protein family it belongs to, because that will help determine or infer the function of that sequence that you are interested in. Or you want to know which domains are there in the sequence of my interest. Okay? So that will be predicted using profile hidden Markov model. So you can go to PFAM, you can input your sequence, and PFAM will predict the protein family that the sequence belongs to, and also the domains, conjugate domains that are present in the sequence. Okay? Okay, so this you will do when you are working on tutorial problems. Yeah, hidden Markov model has been used for transmembrane protein topology prediction. Transmembrane, you know, transmembrane, it spans the membrane. So let's say that this is the membrane cell, and this is outside of the cell. So outside, and this is inside of the cell. Okay. So transmembrane protein, it spans the membrane, so it goes inside and then come outside. Okay. And you would want to know that which residues are outside, which residues are inside, and which are within the membrane. Okay. So that's called topology of the transmembrane. And as I just mentioned, you will say that okay, there are three states. One is outside, the other is within membrane, and the last one is inside, right? But of course, this has been modeled using many more states. So you can see the states in these squares or rectangles. So within membrane, there is a helix core, okay, helical structure by these residues. And then outside, which is non-cytoplasmic side, you have loop, cap, and similarly within the cytoplasmic side, which is inside, you have again loop. So it will loop around. So these are modeled by loop state, and then globular residue. For example, if you look at this one actually, so cap, so how many, so you can see the cap states. So cap, let's look at this. So how many cap residues? This is how many cap residues? 5. Okay, 5. And then loop around, right? So how, what is the minimum residue, the minimum length of loop? No. So you can come here and you can go directly to this cap. So it could be one residue, right? You can make a transition directly here also from this. So just what could be the maximum length of the loop? 10. And then you you can go to globular, or you can directly transition to this. So okay, so 10, but so this, and then so, and then how many are here? Loop? No, 10. Total 20, right? So you can transition. So you can transition to globular, or you can directly transition to loop again. Okay. So 10, and then you can transition here, and then 10 again. So total 20. So loop is variable. So it can even have one residue, or it can have 20 residues. So in between this, okay. So this is all being modeled here. So this you see how these residues, lengths of these residues are being modeled. So modeling variable length, okay. And then you have the helix core. Helix core is also being modeled here, variable length. So what is the minimum length of helix that is being modeled? Minimum 1, 2, 3, and then you make a transition here, okay, 24, 25. So minimum is 5, and maximum is 25. So between 5 and 25, okay. Okay. So, and let's say that okay, we should be, because these are posterior probabilities, so we can sum up these probabilities here within the cytoplasm or inside. So, then you can say that this is the probability of being inside, okay, and then probability of within membrane, and then sum all these probabilities, posterior probabilities, and then that would be probability of being outside. Okay. Okay. So, this was used for predicting the protein topology for a transmembrane gluconate permease 3 from E. coli. Okay. So, you can see actually the posterior probability plot where transmembrane is indicated by green, inside by red, and outside by blue. Okay. So, let us look at the first few nucleotides, sorry, residues, first few residues. So, which has the highest probability, transmembrane, inside or outside? No, yeah, this only outside, blue. Okay. So, which means that the first few residues are outside of the cell. Okay. And then what is the next, from outside to transmembrane to within membrane? So, from this position to this position, which is I think close to whatever it is 40 or so, the residues are within the membrane. Okay. And then what next? Inside of the cell, and then again within membrane, and it goes in this cycle. Actually, the model is confused at some point. For example, if you look here, it is not clear that whether you should say inside or outside. Okay. But it got resolved later on. You can see it is well resolved. So, there are challenges because this is using forward backward algorithm, and what forward backward algorithm does? What it does? Quick, it computes the posterior probability of a hidden state. Okay. So, that is also called posterior decoding. Okay. Okay. So, and this is actually also the same thing, and a new model was developed later on, and the power of hidden Markov model is that it is modular. You can incorporate as many modules or features as you would want to, and it can work wonder. Okay. So, in this model they included the signal peptide which leaves the transmembrane, but later on it is cleaved and mature transmembrane is obtained. Okay. So, they included signal peptide also because there was a cross prediction between signal peptide and transmembrane because they have some shared characteristic. So, as I told before that in order for model to work, you have to show model everything, then model is well informed and it does good. Yeah, it will not generate spurious prediction if you have shown model as much information as you can. So, this is actually the same thing, but it is helping in preventing cross prediction between signal peptide and transmembrane, and this program is called PHOBIUS, which was developed after the TMHMM for predicting transmembrane protein. What is the time? 1:10. So, we will take a break and we will come back at 2. Okay. Okay. Yes.
Actually, we have a few questions not related to the lecture, but for our research, could you help us out?
Yeah, but yeah, you should all have food actually first, and at some point we will discuss.
So, could you give a time to be?
Yeah, I don't know because I will leave maybe tomorrow. It is possible. If not, then contact me on my email. Okay, you have the email. Yeah, so okay, contact me on email and we can meet on Zoom if we are not able to meet. I don't know about the schedule tomorrow, but in case I am not able to, contact and then we will discuss. Okay. Okay. Yeah. Oh yeah, you निकाल लीजिए.
Thank you.